So good evening. I see there are still some participants that are joining us, but I'm going to go ahead and just get started with some quick introductions. It's going to be a few minutes while people are coming in to participate, but we have 2 rounds of introductions tonight, so I still think we should go ahead and try and be timely. First, I want to thank very much all of the participants that are coming on to hear this evening lecture. This is really a continuation of our community lecture series, which we have once a month in the Stem Cell Research Center. You can find information on that on our website. We just finished a fantastic symposium today. This was our 8th annual Stem Cell Research Symposium at the Sue and Bill Gross Stem Cell Research Center. It was a really terrific day of scientific talks, but traditionally at the end of that, we gather with an evening public lecture. Unfortunately, we're still unable to do that in person, but that's okay because we have a terrific speaker lined up for you this evening that will really highlight some of the important things that are going on in terms of stem cell research and regenerative medicine research, not just at the basic science level, but really illustrating what the path is to translation. And this is something that's really critical, particularly in California with the refunding of the California Institute for Regenerative Medicine. As many of you have heard me speak about over the last at least 2 years, this has been a really critical aspect of the initiatives from CIRM to move research out of the bench and to the bedside. Towards that goal, many of you know that we have an alpha stem cell clinic that's based here at UCI, directed by Dr. Daniela Boda. We're really proud of that in terms of both our relationships out to the community and having an impact on regenerative medicine and the capacity to run high quality clinical trials that are making use of cell-based therapeutics or targeting stem cells. Towards that goal, that really comes to what our lecture topic is going to be tonight. So I'm first going to introduce to you Dr. Claire Henchcliffe, who is our new chair of neurology at UCI. She joined UCI just last fall, October of 2020. What a wonderful time to take over this role. And then she is going to introduce for you our main event speaker tonight, Dr. Lorenz, with whom she's collaborated very extensively. Claire is a fantastic addition to UCI. I'm so pleased that she could join us tonight to be able to introduce a little bit about her role in neurology and where we're going on the clinical side of things here. And she's such a great addition because she comes with a tremendous amount of experience. She's previously vice chair at the Weill Cornell Medical College, where she also ran the National Institute of Neurological Disorders funded NeuroNext Clinical Trials Network. She has her MD from Columbia University College of Physicians and Surgeons in New York and her PhD in molecular and cell biology from Oxford. She managed to squeeze in a couple of postgraduate fellowships at Cambridge and at UC Berkeley in her spare time. And so I am delighted to be able to introduce you all to her as our current chair of neurology. With that, Claire, thanks for taking the time to be able to join us tonight. And please go ahead and introduce our main event.
Okay, that was way too generous, Dr. Anderson, but I have to say I'm thrilled. I've been here for 6 months at UC Irvine now, and being able to work with the researchers involved in stem cell research and with the team over at the Alpha Stem Cell Clinic has just made this such an amazing place to join. But I have known the speaker, Dr. Lorenz, for many years now, and I have the distinct pleasure of introducing him. He is truly a preeminent stem cell researcher, and his work has just made enormous strides possible in the stem cell field and in clinical research, and particularly in my specialty of Parkinson's disease. So you already know he's serving as director of the Center for Stem Cell Biology, and he's a member of the Developmental Biology Program at the Memorial Sloan Kettering Cancer Center, as well as being professor of developmental neurobiology at the Weill Cornell School of Medicine in New York City. And that's where I was incredibly privileged to be a part of his team for some years. So just a little bit about Dr. Studer, and I know that you are going to very much enjoy his presentation this evening. Dr. Studer was really among the first researchers to realize the potential of studying stem cells from individual patients to understand human diseases. That's actually how I first met him, I think. It was really exciting for our patients in New York at the time to know that they were contributing to this newest research, and it's really become an important area for drug discovery. Importantly, since a lot of neurological disorders, including Parkinson's, are very much age-related, he and his team developed key strategies to manipulate aging in cells. And that's really important when you're studying diseases that are age-related. And before moving to the more translational part of things and the clinical research that we're going to be hearing more about, I just wanted to mention the sheer importance of his work has been recognized with numerous awards. He's the recipient of the MacArthur Fellowship, the Ogawa Yamanaka Prize, the Jacob Heskel Gabe Award from Brandeis University, and the list just goes on and on. So I really don't have time to list all of his accolades. He's extraordinarily well published and a leader in the field. So I think what has been so compelling for me as a Parkinson's clinician and a Parkinson's researcher is that Dr. Studer's lab really established many of the currently available techniques for turning human stem cells into different types of nerve cells that you would find in the nervous system. And one key cell type is that he really pioneered how to turn stem cells into dopamine-producing nerve cells. And these are the cells that are progressively lost in Parkinson's disease. So, of course, that really opened up the pathways to be able to provide a regenerative medicine approach to Parkinson's disease. And with his major interest and drive in regenerative medicine, Dr. Studer spent many years now leading a multidisciplinary consortium in New York to develop human stem cell-derived dopamine-producing nerve cells for the treatment of Parkinson's. And, with 6 Parkinson's disease specialists in my department, we see day in, day out the sheer scope of challenges faced by people with Parkinson's. And I do believe that Dr. Studer's work is really going to be important in pushing us forward to new and better treatments. Now, the exciting thing is that he recently gained approval by the FDA. And in his role as scientific co-founder of Blue Rock Therapeutics, he now has a first-in-human clinical trial starting up for patients with Parkinson's. The surgical lead is Dr. Vivian Tabar, who's the chair of neurosurgery at Memorial Sloan Kettering. Dr. Harini Sava is my beloved colleague over at New York, and she is leading the neurology team there. But it's really made doubly exciting for us because we are planning to participate as a site in the clinical trial and through the Alpha Stem Cell Clinic. So again, thank you, Dr. Anderson. Thank you to your team for helping us with that. And with that, I will turn over to Dr. Studer. I'm looking forward to your talk and welcome.
Thanks so much, Claire, for this wonderful introduction. And thanks so much also, Eileen, for the invitation to give this special lecture tonight. So I hope you all can see my presentation. Otherwise, please let me know. Now, what I really would like to do today is to get you a laser pointer. What I would like to do today is to give you a little bit of an idea of the journey that we have been on to really try to now move stem cells into the clinic for Parkinson's disease. And as you can see in this picture represented in our recent publication, it was a complicated journey. There were many steps to climb, many challenges to go through. And I will tell you some of those, maybe again as an example for the field in general of how we can bring pluripotent stem cells to the clinic. And as Claire indicated, I also have a hat in co-founding a company called Blue Rock Therapeutics. And both me and Vivian Tabar, which for full disclosure is also my wife, the neurosurgeon involved in these studies, we are scientific co-founders of Blue Rock Therapeutics. Therefore, you should take that into consideration in my presentation, because obviously it might benefit from whatever happens in this trial, also as a partner of Blue Rock Therapeutics. And again, these are my disclosures. But really where I want to start off is obviously Parkinson's disease itself. So as many of you might know, Parkinson's disease is characterized by being a movement disorder. So what that means is that patients have problems in various aspects of movement, such as, for example, their gait. They're often hunched over as shown here. They have small shuffling gaits. A common feature is the shaking, called a tremor in the patients, or when they write, they tend to write smaller and smaller as they go. Now, it's thought that all those movement-related problems are caused by the loss of a very specific cell type, which are these dopamine cells deep in the brain, in a region called the midbrain or the substantia nigra. It's called nigra from Latin because this region actually has this black pigment, which is characteristic for those cells and quite unique, actually. It turns out these cells are quite rare compared to the about 50 billion cells that each of us has in the brain. It's only about 300,000 to 400,000 of those on each side that are actually those specific dopamine neurons that get lost in the disease. It's estimated that once we lose about half of those cells, we start to suffer from symptoms of Parkinson's disease. Now, Parkinson's is a quite common disorder. In fact, it's the second most common neurodegenerative disorders after Alzheimer's disease, affecting roughly about 1 million patients in the US. Now, it's also a big problem economically, and there are estimates now that many tens of billions of dollars are spent on Parkinson's disease directly and indirectly in the economic burden caused by the disease. So very, very critical that you find new and effective treatments. Now, equally important to say, it's not just a disease of the dopamine system. In fact, some of the very earliest symptoms that patients often report, even before they are diagnosed as true Parkinson's disease patients, is that they might lose a sense of smell, that they have certain movements in the sleep called restless leg, or that they suffer from constipation, which is due to the problems in the nervous system of the gut. So it clearly affects multiple systems. It's not random, but it affects a number of systems beyond dopamine neurons in the midbrain. And that's an important point to consider when we develop a dopamine-based cell therapy. So we might actually not be able to fully cure the disease completely, but our dream is to cure the movement disorder aspects of the disease. And again, one area where we definitely need to think hard beyond dopamine neurons are the fact that some of those patients also at later stages of the disease develop cognitive problems, which can also be quite debilitating. So obviously one of the first questions talking about Parkinson's disease is where does it come from? Why do we get Parkinson's disease? Now, one area that we kind of first look on is the genetics. Now, it turns out that actually in most patients, there is not a single gene that is known to cause the disease. More than 90% of the patients, this is basically a sporadic disease, and the environment might play a major role in whether you get the disease or not. However, there is a smaller number of patients where there seem to be genetic factors that can have a major contribution or smaller contributions, alone or in combinations. And what is really interesting from some of those genes that were linked to the disease, we get some of the ideas of what might go wrong in the actual disease. I'm not going to go too much into that, but just to give you a little bit of a flavor, what might be some of the causes and what might be unique why these dopamine cells die, is one fact is that these dopamine cells are unusually a large cell type. So this is actually just a scientist in Japan who traced a single dopamine nerve cell in the mouse. And you see here is one process that goes out of the cell body. The cell body would be far away. And then it branches into this huge network of cells. And it's estimated, this is a mouse neuron, it's estimated in humans that one dopaminergic cell has a branching tree of about 4 basically yards in length. So that's crazy if you think in cell body. That's a single cell that needs to sustain that process, that tree of processes, and needs to do that for the whole lifespan, because those cells are born very early when the body is still as an embryo. They are born very, very early, and they have to last, hopefully, for the whole lifespan of whatever it is, 80 or 90 or 100 years. So therefore, these cells have a huge energy demand, and in fact, some of the genes that seem to be linked to Parkinson's disease affect the energy production sites in the cell, which are called the mitochondria. Now, another area where things maybe can go wrong is in controlling the protein function, particularly the protein aggregation, because the diagnostic feature of the disease is that patients develop a structure called Lewy bodies, which are an accumulation of proteins, including particularly a protein called alpha-synuclein. That's very interesting because that might be actually a common theme in some of the neurodegenerative disorders, not only in Parkinson's disease, where alpha-synuclein is accumulating. In other diseases such as Alzheimer's disease, there are other proteins that are accumulating and are basically aggregating and seem to cause damage in specific cell types. And so this is all very interesting, and we made a lot of progress figuring out the genes, the mechanism. But if you're a patient, do you really care about those issues directly? You really care about novel treatments. And so the sad story there is actually right now, none of those insights has led to any new disease-modifying therapy yet. We have a lot of hopes for the future, but right now, we don't really have any disease-modifying therapies at all. Now, the therapies we do have is that we try to just simply replace the product that comes from those dopamine nerve cells, which is dopamine itself. And so you can use the precursor called levodopa, and together with another drug, give it systemically to the body. And this can be quite effective. We can really replace dopamine function often for several years in a patient. It was a revolution about 50 years ago where some of the patients could get off the wheelchair and finally move again after many years of being nearly completely frozen. The problem is as the disease progresses, and it's obviously a progressive disease, this works less and less effective because the cells are still involved in the conversion of this precursor into actual dopamine getting properly released. Now, another approach trying to treat diseases by just simply bypassing the need of dopamine cells by stimulating specific parts of the brain. And that can be quite effective for some of the symptoms, such as the shaking, the tremor I showed you, but it's not suitable for all the patients. And it has certain features that make it, again, sometimes a little bit cumbersome, or also can, in some cases, maybe make it more difficult for patients in certain specific symptoms, such as, for example, in the context of speech. Now, these are the currently approved therapies that basically are FDA approved and the therapies that can be applied to patients today. Now, the experimental therapies is on the one hand gene therapy, where we try to introduce genes that then would maybe enhance the function of the remaining dopamine cells, or have other cells in the brain try to take on the function of dopamine cells. These are some of the main strategies being tested. The problem with that is, again, if obviously the remaining cells are getting continuously undergoing this degenerative process, this might also be quite challenging. So what we therefore propose is maybe, why don't we just simply put new dopamine cells back? And that's, again, obviously the main focus of today's presentation. Now, we were not the first to think about that. And in fact, there's a long history of trying to replace dopamine cells. And in the past, it was particularly attempted by actually isolating dopamine cells from the region where they are first born. And I mentioned that they are born actually in the embryonic stage, about 6 to 10 weeks after conception. As you can imagine, this is quite cumbersome. It has a lot of ethical and logistical issues, but still that's something that has been tried. Now, where we think the future is doing that with cells such as pluripotent stem cells. I'm going to talk more about what I mean by that, but by a cell that can really self-renew long-term and potentially can turn into any cell type of the body, including, obviously, dopamine nerve cells. So this is the area where we want to go. But one point I want to make here, again, in a public setting, is that we have to be also very careful of what seems to be not a source of dopamine cells, but it is often proposed as such currently already. If you're actually going to go and use some of the currently search engines and type in stem cells and treatments and Parkinson's disease, often among the very first hits you get is companies that try to propose that they already have a stem cell therapy for you. It doesn't really matter whether you have Parkinson's disease, whether you have Alzheimer's disease, autism, the same cells can do it. And so obviously that's clearly quite problematic from our perspective. I'm not allowed to tell you which companies there are because they're very litigious companies. But just to make the point, you have to be very careful. And biologically, we know that those cells don't turn into dopamine cells ever and actually don't even survive in the body long term. So therefore, patients have to be very careful when we talk about stem cells. Stem cells are not stem cells. Very important that they talk with their neurologists and scientists to really understand what they get into if they want to have such a treatment. Because not only might it not work, but there were examples, for example, where stem cells were used to try disorders of the eye, which is again a very important area. I think Sally Temple spoke to this community last year. And actually there were cases where these unproven cells are caused actually the loss of eyesight. And so again, it can be not only not effective, but actually potentially dangerous. Now, what we learned from the fetal cell studies is kind of a mixed picture. We knew that actually dopamine cells can survive in the brain of a Parkinson's patient and that they can produce dopamine. But this therapy was tried in about 300 patients worldwide, and the results were actually quite mixed. Not only were the results not always completely satisfactory, but some patients even showed certain side effects, which are called dyskinesia, which is kind of uncontrolled movement. In Parkinson's, often you have not enough movement, but in this case, you have kind of overabundant but poorly controlled movement. So this was kind of disappointing, again, that fetal tissues seem to be not providing an immediate solution, but they actually still gave us some hints that there might be hope. Because actually what was done beyond this initial larger scale placebo-controlled studies, some patients were followed much longer than the 1 year of follow-up that led to these mixed results. But some of those patients were followed 10 years, 15 years, up to 20 years. And at least a subset of the patients seemed to have quite remarkable benefit. And in some cases, actually could stop their levodopa treatment, which is nearly unheard of in such a progressive disease. So this would again suggest that potentially this could work if you did it in the right patients, and maybe you need enough cells, maybe there are other factors to consider. And in fact, in Europe, they have started again a trial to exactly figure out which factors could be improved in fetal cells to maybe make that more efficacious than in the past. Now, what we've learned from the fetal studies on the one hand is clearly that patient selection is going to be quite important. I'm going to come back to that for our own trial. We also know that we probably need to wait quite long before you're going to see the full potential effect of those cells, not only 1 year as in the past studies, but up to 2 to 3 years, where they basically develop their full potential. We got some insights about the need of immunosuppression, which is again an important area. And we know that basically cells that are not matched, so they come from another individual, they can survive for a very long time. But it's the idea that maybe we still need to have transient immunosuppression initially. But the point is that cells are shown here. This is a brain of a patient that died due to another cause 24 years after receiving this treatment. And this brown stuff here is basically dopaminergic fibers coming from the graft 24 years later. 23 years of those 24 years without any immunosuppression. And finally, again, we also learned maybe the challenge that it might not fix some of the other non-movement related symptoms such as constipation, smell, or cognitive function, for which we might need to have parallel approaches in the future. So again, but maybe the most important lesson was also that fetal cells are not going to be the future. For all the ethical, logistical issues, it was very, very cumbersome. Even the trials today, they were struggling over several years to just get about 10 patients ever transplanted with such tissue. So what we really want to have is a stem cell-based approach that really could make the difference. Basically, the goal is to replace lost function and bring function actually back. In this case, that we have new dopamine neurons that can bring this function back. And they might do more than just secreting dopamine, because they can actually hook up in the brain properly. And also we think that if this really works, a one-time surgery might give us the long-term benefit for 10 years, or what I just showed you before, maybe up to 25 years. And therefore, if you can do that as a kind of an off-the-shelf treatment, where you can make a lot of stem cell-derived dopamine cells, maybe this actually could be done at scale and quite cost-effective. Now, this was quite a long journey for myself to actually go through that. And just to give you a little bit of a flavor, again, this didn't happen yesterday. I was actually, my own journey started, I think, more than 20 years ago. As a medical student, I was involved in some of those fetal transplantation studies and realized again that fetal cells are clearly not the solution, even though they are quite interesting. And I thought initially neural stem cells are the solution. So stem cells out of the brain, isolated in addition, converted into dopamine cells. But actually, even though we had some interesting publications on that, this seemed to not really give us a truly unlimited supply. But it was an area where actually I met my partner in crime for the rest of the story, which is again Vivian Tabor, basically to then take that to the next level, which are embryonic stem cells first in the mouse, and where we quickly actually were able to make conditions where we could treat at least mice and rats with mouse-derived stem cells. Now, obviously, these were exciting data, and in fact, these were some of the data that led to the first excitement about starting efforts like CERN, but doing that now always with human cells. And at that time, we actually had human cells, human embryonic stem cells, from people like Jamie Thompson, who actually figured out conditions to get unlimited numbers of human embryonic stem cells. And these cells have, in their nature, the potential to turn into any cell type of the body. As you all know, we have now another option as well. Thanks to Shinya Yamanaka, who got the Nobel Prize for his work, we can also start with adult cells, convert those cells back with 3 factors called the Yamanaka factors, back to a cell that's pretty much indistinguishable from an embryonic stem cells, but has just started from an adult-like cell, can be matched to a given patient, and again, can give us rise to all those various cell types. So the challenge in the field really has been, how do we do that reliably? And this was, again, a long process, how to control cell fate in a dish. And again, to just give you a little bit of an insight, kind of a look under the hood of how to do that, we developed a lot of those protocols, as Claire mentioned, where we can just simply take 2 small chemicals, feed it to the stem cells under the right conditions, and the cells now go down the path of development where they would have all these various options, but in the presence of those molecules, they're only going to make brain cells. With another set of small molecules, we can actually tell them which part of the brain they make, whether they make the forebrain, the midbrain, or the spinal cord. It's a little bit like a zip code system where we can give the right zip code with the right factor to pattern those emerging brain-like cells. Now we can perfect that, and we did that for the dopamine neurons over a period of about 10 years, where we have now a very detailed protocol with many of those factors to make bona fide dopamine neurons that seem to be pretty much indistinguishable from the cells that would be born normally during development. So this is a very powerful approach. And again, already about 10 years ago, we made those key steps that basically enabled that for our own group, and actually now for many groups across the globe who want to develop similar approaches. By learning how to do that for dopamine cells, we actually also learned to do that for many other cell types. So the same logic, the same zip code strategy that I mentioned to you, you can now use to make pretty much all the cell types of the brain or of the peripheral nervous system. Not all of them are perfect yet, but I would expect within a couple of years, we're going to have the building blocks to basically make you any cell type of demand and at scale. What you can then do is, what you showed in the study many years back, you can then take, for example, those dopamine cells completely generated in a dish and inject them into a mouse, rat, or a monkey model of Parkinson's disease and show that these cells not only survive, but they seem to improve the behavioral deficits associated with the Parkinsonian symptoms. Now, in the mouse and in the rat, we use very simple assays, such as this, for example, rotation assays. This is a sped-up movie where a mouse or a rat is basically has removed dopamine cells on one side of the brain. It's stimulated, only one side responds, but after transplanting them on the cell that the dopamine cells are missing, with a new complement of human stem cell-derived dopamine neurons, the animal stops rotating. Another assay is shown here, where an animal can no longer initiate the movement on one side, but this basically can do so again after the cells basically have taken on function in the brain. So you see it doesn't drag the paw, but then it starts initiating movements. And that's a bit closer to what we see in Parkinson's patients, where patients often have exactly this problem of properly initiate movement. In Parkinson's, you're not paralyzed to make movements, but you have a lot of problems to initiate and properly control the movement, which is kind of mimicked in this assay. As a scientist, we were also curious to know how exactly these cells work. And we could actually show, using kind of a fancy technique called optogenetics, that we can put cells into the brain, have these animals recover, like I showed in the previous movies, but then we can actually switch off the cells in an animal as it moves. And what that shows us, literally, is like a light switch. We can show that all the benefits, all the improvements that we showed in the previous slides go away, showing that actually the way the cells function is properly have neuronal activity. That's absolutely essential. And actually neuronal activity gets integrated into the brain itself. And so that's different from other stem cell therapies where we try to rescue the other cells in the brain. These are actual cells that take up shop in the brain, that function, that fire, and that's critical for their role. And therefore, again, transplanted cells need to be electrically active and they need to make functional connections. Now, thinking how we translate that for the patients, that's really where we got very closely to work with Claire, was basically got funding from the New York State to actually do that. All the way from planning how you might do that, from these very exciting scientific studies, all the way to the bedside. And so we got a big award. And again, I will show you some of the fruits that basically were harvested from this effort. And I just want to give you a very sad update personally from that. Unlike you guys in California, who are very lucky, just got funded again. We just got the message literally last week that this very successful program that enabled us to move all the way to a clinical trial was actually stopped. And again, we are very sad about that, and we'll still try to lobby maybe to reinstate that, but it was a very sad and unfortunate situation in New York. But so one of the first steps we had to do in this process is actually make that now not only in our lab, but make it a clinically compatible version of dopamine cells. So we need to use very special reagents that the FDA accepts, that they are safe enough for use in patients. I'm not going to go into the details of that because they were just published in a recent paper. For any of the scientists and the audience are really interested in that, they can read in more detail. But some of the key steps is that we had to make the process even more precise and robust. We had to use, as I mentioned, clinical grade reagents, and we had to make sure that we make the right cells, but also that it can avoid any of the potential, what we call off-target cells. And we and others have in the past shown the number of the wrong cells you might get in addition to the right cells. Some of the, for example, called serotonin nerve cells, which actually are present in large numbers in fetal cells. And we think they were some of the culprits in causing some of the side effects of fetal grafts. So you don't want to have those guys. There are a number of other cells that we also want to avoid and that we show now with our new protocols we can avoid. So with those new improvements in hand, we were now ready to manufacture the cells. So no longer our small lab, but doing that in an actual clinical grade facility and making now billions of cells. Because our goal is to not just make cells enough for one patient, but to make a very, very big stock of cells. That stock can be used for hopefully treating many patients, but also that's the exact same stock that we're going to use to now treat many, many mice, rats, and monkeys again. Because that gives us then the confidence that those cells have been gone through all the rigmaroles to show that they're functional and safe, and from the exact same lot, we're going to treat the patients. So for making that possible, we had to figure out how we can freeze nerve cells. As you can imagine, that's not completely trivial because they have some processes already and took us quite a bit of time to do that. But by now we perfected that technique as well. And so we now tested the cells. Again, I'm not going to go into the markers that you need to do to show that basically pretty much all the cells have the right markers. And for example, none of the stem cell markers are left. The idea is really, thinking in very simple terms, is you want to understand what's in the vial. You want to ideally understand it similar to the way we understand a drug, where you know exactly the composition of each chemical. You want to understand the composition of each cell that's in our product. So the next step then really was going from this step of making the cells, shown here, climbing up a few steps, to asking, no, what is actually now required by the FDA to move through the clinical door here? And so what was required is a huge number of clinical tests with those cells we've produced. So those tests include studies that you'd call efficacy study, show that the cells still work in the right animal model. But particularly the FDA is concerned about safety studies, showing that the cells go to the right place, that they are primarily obviously in the brain, they don't go to other organs and do potentially havoc at the wrong place, that they are not having any toxic effects on any organ system, and most importantly, that they don't form any tumors. And again, we went through all of that in a very large number of animals. And again, I just show you some of the highlights of what you see. And what you see is that, for example, if you look where the cells go, you find that we have only surviving grafts and no abnormal tissue, that the cells do not get sick, so there's no obvious sign of toxicity or morbidity, and that after 6 months, we cannot really detect any cells in any other place than the brain. So these are all good signs showing that the cells follow the criteria we expect for their distribution, their toxicity. Now, the other key point is obviously tumorigenicity. So what you do is you have to actually inject the stem cells themselves, which you know should form tumors in those animals, which are shown here, some of those weird structures, which are called teratomas, which are a wild mixture of tissues. Then we were basically asked to put in as many cells as we can stick into the brain of our dopamine cells and show that even if we do that, the cells don't form any tumors. And so again, we had to test that in many animals to make sure that this is safe. And finally, something we had to learn, we actually didn't realize as much before we did those very extensive studies of up to a year of transplantation studies in those various animal models, is we had to basically explain what happens to the cells themselves in great detail. Not only do they survive, are they dopamine cells, but something we noticed is that actually the graft over time does spread out a little bit. And first, we were nearly worried, could it mean that the graft grows? And again, that would be maybe something you might not want to see if the cells were to proliferate. But what we actually found, it's not that the cells divide in the brain, but what they do is actually just simply, they differentiate into the dopamine cells. When they do that, they need a little bit more space, they spread out. And what actually happens is that the density of the cells goes dramatically down, which correlates exactly to the size of the cells occupied within the brain and intermingle nicely, integrate in the brain. So that's again one of the lessons we've learned from these studies, to know how they really integrate over very long periods of time. Now what they do in the brain, we saw in the efficacy study, they not only survive, is shown, for example, here in this area, in an animal that was completely devoid of dopamine cells and dopamine innervation on one side of the brain, but they actually re-innervate pretty much the whole region where the cells need to innervate, is called the striatum. And so that was again very exciting to see that we can get pretty much full re-innervation, and therefore we can also see pretty much full recovery in some of those behavioral assays in large numbers of male and female, in this case rats. And you see then these beautiful, very large cell bodies interspersed, again spread out within the brain, as I mentioned before, and making many dopaminergic fibers very far away from the actual graft core. Now this leads us then to the next important question, which is really now the actual clinical trial. And again, there obviously we relied a lot again on Claire and Harini in guiding us on that. How do you design a phase 1 to A type study? And what are the primary objectives? Now this is a first in human study, so the primary goal has to be to look at the safety of those cells, how they are tolerated in the brain, and is it really feasible to do that in a Parkinson's patients of the right type? And so what we decided, we want to look at those readouts after 1 year of injection. Now the secondary endpoints of that study is that not only do we want to see that these cells are safe, but that they can survive, and that there is signs of actually that they can produce dopamine, for example, by certain imaging techniques called PET imaging. And we also want to see, even though there's going to be a small number of patients, is there any sign of clinical improvement, again, at 1 year, but you also want to go later at 2 years and follow these patients actually for up to 15 years, possibly for the rest of their life, to really make sure, again, that we have good safety measures, but also how these grafts develop. Because then one of the lessons of the fetal studies, if you remember, was that these cells develop and continue to mature similar to they would do during development over a period of up to 3 years. And again, these are the number of endpoints that we're going to measure. Now, to give you a little bit more details on actually the clinical protocol. So the idea is this is going to be a single-site open label study. And again, we have basically recruitment in the US, and we hope again, going to hopefully have recruitment in California as well. And we also just recently got the permission to do that in Canada. Now, the first trial will be 10 patients. And the way we planned that initially was that we would select patients of moderate to severe stage of the disease. And that's, again, an important consideration. You cannot really do that in very mild patients because it's a first-in-human study that has certain risk, has never been done before. But you might also not want to go to the most severe patients because based on the fetal studies, they might have less of a chance to benefit fully from the approach. Now, what I marked here in red is in the discussion with the FDA, they pushed us a little bit further towards the advanced forms of Parkinson's disease, again, with the argument that this is a first-in-human study. And similarly, they pushed us to a slightly older patient population than what we think is ideal. But again, that's the patients that we're going to move forward with. There are other criteria that the patients have to be at a certain stage in the disease, at least 5 to 10 years into the disease, which is very important, again, to make sure that we have the proper diagnosis. And we also kind of know that how the patients progress, because there can be some variability between patients. There are certain exclusion criteria. As I mentioned, we don't think we can treat dementia, and demented patients obviously would be much more difficult to cooperate in such a context, and a number of other morbidities that might be not ideal for such a first-in-human study. Another interesting question is, how do we actually do that with regard to dosing? So the way we thought about that is we actually know quite well how many dopamine cells are normally there in the brain. If you paid attention, I told you there are about 300,000 to 400,000 in the substantia nigra, the midbrain of a normal individual. And actually, we know from our transplantation studies how many of our cells do survive. And so we can now calculate that, for example, for our high dose that we're going to use in those patients, the 2.7 million going to be injected on each side in 3 tracts and 3 deposits in the target region should give us exactly about the number of cells that would correspond to a near normal complement of dopamine neurons, about 300,000. The low dose, which is going to be the initial 5 patients, is going to be 0.9 million patients, and we calculate that would give us about 100,000 new dopamine neurons. And if you remember again, if you lose half of the cells, 150,000 cells, you get Parkinson's symptoms. So if we give you now 100,000 back, maybe that gets you over the hump to get some benefit. And in fact, if you look in the fetal grafting studies, some of the patients that actually had clinical benefit, they needed to have just about those 100,000 cells when they came to autopsy. But the way we're going to do that clinically is we're going to inject the cells into the brain in a single session in a bilateral way, so on both sides of the brain, and again, doing about 3 tracts on each side. And again, with regard to safety, it's interesting to consider, because the FDA asks us to inject as many cells as we can stuff in a rat brain or mouse brain. We actually inject some of those animals up to 400,000 cells in this very tiny mouse or rat brain. Now, in the much larger brain, our low dose is actually going to be only 900,000. There's only 2 times the dose in a mouse or in a rat. So clearly, we have a huge safety margin in this context, but at the same time, we still hope to have a chance for these patients to benefit with the arguments I just made before, that you actually get numbers that in fetal grafts seem to be clinically relevant. Now, these patients will be immunosuppressed, at least transient. They alluded to that a little bit. The brain is unusual. So it's not like the kidney where brain cells get simply rejected if they are mismatched. But there is still some initial immune response against the graft. So the compromise is to do a transient immune suppression, which should give very little side effects to the patients, because some of those drugs, they have long-term risks, but less so shorter-term risks. And again, the study location currently is Weill Cornell and Sloan Kettering. And again, we hope, obviously, with Claire now, that UCI will be part of that effort for the patient to basically be followed up. Just again, to make it very clear, so these cells have to be directly injected into the brain. And for that, we need a neurosurgeon to do that. And again, 3 tracts on each side. And the way we do that is under basic imaging control. These are the so-called MRI machines that many of you probably know about. But in this case, the MRI is literally in the operation theater directly. So you can actually do that. When you inject the cells, you can see where this needle goes. You can plan it such that there is minimal risk to hit any blood vessels, maximum chance to hit the target. And you can actually, again, follow that in kind of real time. And this is one such example where in this case, we show injection of a drug using the same injection device. You can see that it goes to the right place. And to have this setup was actually quite critical when we discussed with the FDA, the point that we want to inject already in the very first patient on both sides of the brain, because the concerns that you have by doing both sides is, what if you cause a bleeding? You would not want to have bleeding on both sides of the brain. But because we can check very carefully with that imaging directly in the OR, we can make sure that there was no bleeding on the side where we injected the cells before we move to the other side to deliver the remaining cells. And so again, as already mentioned in the introduction, we were very excited that finally, after again a long journey, we came to the stage where we got the clearance of the FDA to start this trial. And actually just even more recent, we got also the clearance to start a trial in Canada with that, and the trial is now open. And again, if you want to have more information, you can obviously, if you are at UCI, I'm sure you can talk to Claire directly. We also set up an email address for people interested, again, in this trial. And again, it's led by Claire and by Harini. Here is the PI here at Weill Cornell. And the surgery will be done at Sloan Kettering by Vivian. And again, this is a very exciting area where we're now just getting to the stage to actually perform some of those very, very first injections. Now, just to give you a little bit of a broader perspective, again, zooming out. So we are not the only one thinking about that. And we are actually, from very early on, we planned that as kind of a coordinated effort across the groups. So when we published this Nature paper in 2011, many groups wanted to try, because finally we cracked the way to make the right dopamine cells. And so there are at least groups in the US, in Europe, and actually in Japan who try to do that. And we have this global consortium, which is called G-Force PD, to try to coordinate some of those efforts. For example, trying to figure out which is the best patient populations and regularly update each other about some of the challenges in moving this project forward. And in fact, there have been 2 trials that have been started in parallel that in this case are a little bit different, because they are actually still not what we would think an off-the-shelf trial. So they were kind of more kind of a first attempt. In one case, it's literally an N of 1 patient that was transplanted with his own stem cells based on compassionate use. So you have a little bit less stringent regulation, because not really a real clinical trial, it's a compassionate use application of stem cells. And there's some data that at least it seemed to be safe in this initial period of time. Then in Japan, there is Jun Takahashi, who has a trial that will be for 7 patients. Again, not really off the shelf, so they have to produce the cells for each patient. They are not fully matched in their case. It's a single stem cell line, but they cannot yet cryopreserve, so work really with frozen cells. So for each patient to have freeze the cells from an early stage, grow them up, test them, then inject in the patients. The reason why I stress this point is it's not really an academic effort to say, okay, this is really a little bit different. We think it's really important, because if you want to really ever treat many patients, it's absolutely essential. We think that you think of it like a drug. You can make a lot of it, you can qualify it, you can use it again, so you don't have constant variability every time you produce the cells. Again, I could go on about many, many technical challenges, how we might want to make this an even more efficacious approach. We have ways we can maybe have different stem cell types, even better dopamines in the future. But obviously, for this clinical trial, one of the next steps is how you move that then to even beyond this phase 1 study that hopefully is going to show that this is going to be safe and efficacious. There are many, many questions that we are happy to further discuss. And again, one question on the mind that is often asked that I put up here, there are many more questions, is obviously how to best move that forward if you have now a larger population. So our first population doesn't have a so-called placebo control. As you might know, again, the so-called double-blinded studies, where for drugs that's pretty much routine, know that the patient doesn't really know whether it gets the placebo or not. You might know that from the recent COVID trials of the vaccines. Now, that was very, very important in 30,000 patients for Pfizer and for other Moderna vaccines, that it was shown in a blinded way that these basic vaccines are efficacious to really give us trust in the results. Now, the challenge in surgery is a little bit that this is not so easy to do. And there are, in fact, many reasons why you might want to do it, because in the past, again, sometimes surgeons do certain treatments There's a long history even before fetal studies where they, for example, try to inject different dopamine-like cells that come from the adrenal gland. That was done for many, many years. And in post-hoc studies was shown was not efficacious at all. It was never really fully tested clinically. Surgeons just went ahead, did it. So it was really important to really kind of figure out having a better clinical trial to prevent that. It might be required for regulatory approval if this becomes a routine treatment. And obviously, in Parkinson's patients, it's not unheard of that there is a significant placebo effect, at least transient. And again, it might also reveal unexpected side effects, which was the case in some of those placebo-controlled trials with fetal tissue. On the other hand, with surgery, it's not just minimal risk to get surgery if you don't get any cells. So this would be patients who would have to agree to undergo surgery, to get even a burr hole into their skull, and potentially to even get immunosuppression. Otherwise, how do you really properly control for the patient? And that's clearly significant risk. Furthermore, these patients might then be not able to participate in any other trial. They would have to stay on this placebo for the whole period of the trial. And what we really hope, and again, maybe that's a pipe dream, is if our effect is really very obvious, and that was often happens, for example, for rare diseases, where you have only a few patients, you have a genetic disease, you have a treatment that's very, very efficacious already in early studies. The FDA basically says it's so obvious that you don't really need to have a proper placebo in this case, but you can basically have other control groups to get actual approval. And then finally, the final point I tried to make here is that for fetal studies, maybe if you use it prematurely, the way it maybe was done in fetal studies, you might also have a long-lasting setback on the field. And the interesting snippet there is we actually go back, flash back in history to 2001 and look at the front page of the New York Times. You find one area actually right here highlighted by Gina Colada, a report that was really quite detrimental to the fetal grafting field because the way it was presented, not only again, did it not really reach the primary endpoint, but it supposedly has this disastrous side effects. And again, you can talk to Claire if you want to figure out how disastrous the side effects were. They were clearly a problem. It's something we think we can avoid in the current cell products. I mentioned, for example, the serotonin nerve cells might be the cause of it. They are not present in our current cell product. But again, it was presented in a way that it was really very difficult for the field to move forward for many, many years, because every neurologist who would read the New York Times says this is clearly not going to work. It's even going to endanger the patients. In hindsight, again, we think, at least in part, it was done because we didn't really know enough about the patients, about the approach to really start a placebo-controlled trial that clearly endangers risk for a placebo group. So this leads me then to really my conclusions here, and hopefully leading into a further discussion with questions from the audience. So we think that dopamine cell replacement therapy is now finally ready, using human stem cells to be tested clinically. And in fact, again, I made the point that we are now ready to move forward with our study. We have clinical clearance. We think, on our side at least, it's very important that we can do that also off the shelf to bring eventually this therapy to the broader community, to not just have one-by-one patients where the therapy might cost $1 million or more to actually make that work, and again, might have suffer from variability to patient. On the other hand, again, there are a lot of remaining questions. I mentioned one very specific one: how are we going to control for such a treatment in clinical studies? Are we going to continuously need even this transient immune suppression? There are pros and cons for that. We have some creative ideas how we can use cells that might actually not need that at all. You can engineer the cells with fancy CRISPR techniques, so they are kind of shielded. They're like stealth cells that don't get recognized by the immune system. And we have such technology in the company that we co-founded called Durox Therapeutics that in a future study maybe could be used without any immune suppression. And again, also the question is what's exactly the best source long-term. The other point that hopefully you got a clear flavor from my presentation, these things take a really long time. So it took us about 10 years to figure out the recipe to make the dopamine cells. It took us another 10 years from the recipe to actually now getting the approval to go into the clinic. So how can we accelerate that for future trials in Parkinson's disease or for other disorders? And related to that, I alluded to that a little bit too. What about other symptoms of the disease? can we replace all the cell types? Can we treat all the symptoms? Can we treat all the diseases? I mentioned to you a little bit the tree that we're starting to build more and more. So we clearly can make the cells in a similar manner to dopamine cells, but which of those cells can treat which disease, and which lessons from our effort here in Parkinson's can we apply to all the diseases? But also to bring it back to Parkinson's disease, one of my dreams is that we can use approaches to treat the other symptoms of Parkinson's disease as well, all the way from efforts that I'm happy to talk about another time. There's some really, really exciting data in genetic disorders where we can fix the nervous system in the gut. I mentioned the nervous system in the gut causes the very severe constipation in Parkinson's disease. Now, this might be not the first symptom you might want to try to treat, but in the future, why not if you can do it safely? But maybe more importantly, again, we have some really intriguing ideas how maybe also using different cells, not dopamine cells, but cells that might disease modify and potentially treat some of the cognitive related symptoms in the future. But those are right now still a bit the pipe dreams, another 5 to 10 years before those are ready. But I think, again, we hope that our efforts in Parkinson's is really pave the road to some of those approaches in Parkinson's and beyond. Last but not least, I want to really acknowledge the team that really made that possible. In my own lab, there are many, many people involved in many aspects. I just highlight some of them for the Parkinson's project. Particularly important is Tae-Won Kim and So-Yeon Koo, that were really the leaders in the field together. There is Jin-Young Piao, who is in actually Vivian Tabar's lab, again, my partner in crime I mentioned for many years, now working on that. Now, it takes a whole village to really bring it to the clinic. I mentioned some of the people here from our NISTEM consortium, and again, some of the key people here mentioned for the actual clinical trial, again, that is now open and hopefully moving forward to quickly get us through those 10 patients and giving us important insights. I think I'm going to stop here and hope we will have questions from the audience that I will also be very happy to answer.
So I just want to start, Lorenz, by thanking you for a really wonderful lecture this evening. We have a couple, and not actually a fair few number of questions coming in. I want to start with two of the sort of broader questions that we could ask, just partly in the interest of time. One of them, while you alluded to the unfortunate demise of NISTEM just recently, within the last week or so, and to the great fortune that we had in California of having CIRM refunded by ballot initiative. And you and I talked a little bit about that earlier today, the differences between the systems. But I wonder for our audience tonight, if you would just say a few words about how NISTEM impacted these studies over time. What was the impact of having that source of funding in terms of bringing this vision for your research all the way from what you first saw during your medical training in the early days of trials for Parkinson's disease with fetal tissue on up through where you see this going in the next year or so even?
No, you're happy to do that because it's really was absolutely crucial on this journey, and I was like the crucial final, close to final step, and in a number of ways. So clearly we could do a lot of academic work in trying to make some of those dopamine cells, and we did that with NIH funding and other sources. But what happens really very, very often at that stage, you get to this what we call the valley of death. So you have interesting data, but at that data, no drug company is going to be interested in funding your trial or actually giving you the means to actually get to the stage where you can do a trial. So it was absolutely critical to have, in this case, state funding to getting us through that process. And they were actually very stringent. It was a very competitive process. I think they had 3 projects initially funded, including projects, literally all of those 3 initial projects actually now go into clinical trials, one by Steve Goldman and one by Sally Temple in the eye, and the other one again with regard to oligodendrocytes and remyelination. So it was a huge success story. And the success story also went such that it allowed us to start Blue Rock Therapeutics, because we got into the step where now actually there was interest from industrial partners, and there was investment that led to this company now sold for nearly $1 billion now to Bayer Therapeutics. So it became a huge success. And actually, this company now is big headquarters, the whole neural headquarters in New York State itself. So New York got the benefit having a clinical trial happening there, had the benefit of all the research and the training of many people who were involved in the study. And that's the benefit of actually jobs within the state. And so again, I find it a very short-sighted and very sad that this doesn't continue right now, and again might prevent other of those many examples. Now I mentioned that 3 of cells that we have now ready to go. We need again funding for this value of this. New companies that would then go beyond that, hopefully again would support that. And so that's why again, I think California was very visionary. Now is the time to do it. You can say maybe last time it was a little bit early. Many people were disappointed that we don't have the treatments yet, but now we actually built the tools. Now is the time to invest and actually again get many of those therapies the chance to make a difference for the patients.
Yeah, that's a fantastic summary, really. And I think it's really interesting because, as you know, Sally Temple spoke here last year, and Steve Goldman, of course, the 3 of you are all really focused in central nervous system diseases. And this is such a challenge, particularly in terms of the Valley of Death and what's the amount of time that it takes to understand the basic system and really move on through to clinical translation. So many in our audience will realize, but not everyone, that 10 years is actually pretty super fast in terms of progressing. And it's without this kind of level of funding and concerted, stable funding, it's really disruptive. It's impossible to make that amount of progress. So that was a great description. I'm going to jump down to a different question. Everyone who's asking questions in the audience, please forgive me, just because I think these 2 are of the broadest interest. Mitra Hushman, who in full disclosure is newly appointed to CERM and is a UCI grad, a PhD student from UCI, asks, I think, a question that we get commonly from the lay audience. And I think there's often a little bit of a misperception. So I think it's important to get out there. She asks, in your interactions with the FDA, did you feel that they were fair in their data requirements for clearance of the clinical trial? How do you think the FDA is in terms of how they take in clinical trials? Are they more strict with cell therapies than with other other therapeutics like small molecules, for example? And maybe just a sense of the nature of your interactions, because I think people in the patient community often perceive or have this idea that the FDA is an adversary. And I'm curious what your experience has been, because that was not my experience in terms of interacting with the FDA.
No, I think that's clearly not the case. And they are very much want to be a partner in getting these therapies to the clinic. I think the challenge initially was that everyone learns how to do it. So in our case, again, we really try to literally replace the nervous system, at least aspects of the nervous system. These are cells. They're going to be there for the rest of the life of the patient. You cannot take them out. And so the question is kind of what level of stringency do you need to apply for that? Now, what hurdles do you need to jump through? And I think we need to learn about that. And I think it's maybe appropriate at the beginning that they put very high hurdles up until we have more data. And I think that was our impression, that we really need to learn together what's really the best way to regulate that. And again, I mentioned this one example where we basically had to figure out exactly, which delayed us a little bit, know how these cells spread out within the graft and how they actually apparently seem to look like as if the cells grow. Even so at the end, they would not really proliferate, they wouldn't make more cells, nothing like a tumor at all. They were just simply doing the normal thing. But nobody had seen that normal thing, neither the FDA. And so what we found actually maybe even more challenging than necessarily the regulatory agencies on our side was also to find the right partners that the FDA requires. So there's a whole group of so-called contract research organizations now that the FDA requires, that you cannot just do all the studies yourself. You want to have an independent group that does that, assesses it so that you are not influencing the results. And often there is still a shortage of people that have the expertise of doing that. And that I found, again, actually something that actually delayed us a little bit more than I would have expected to really find a pathologist. Pathologists hadn't seen such grafts in the brain. So how did you describe that? And so the terminology was not properly found. I think all these issues are kind of the challenges of doing something first. So I think it's going to get much, much easier in the future. Both on the FDA, they're going to be much more clearer guidelines how to do that, because the FDA, they are clearly partners, but they're not going to give you a menu, say you have to do that. You have to propose what you're going to do. And so again, it's a force and back process where we both had to learn. I think the regulatory agents had to learn what we want to do. We had to learn from them how they want to have it regulated. But it's clearly a partnership, and I think it's going to be an essential partnership going forward.
Yeah, I couldn't agree more. And I think that's really a terrific summary. I have a number of questions that have come in from the audience. Unfortunately, we have almost none that are overlapping. Sometimes I can take and compile a few together, but you've got a lot of distinct ones here. So I'm just going to go through in order. Charmaine Soko says, hey, that was a great talk. She has two questions. One, she's wondering whether the cells being injected are homogeneous, like how similar they are to one another. And the second, a little bit sophisticated for some of our audience, is whether it might be possible to use something like CRISPR-Cas9 gene editing in a therapeutic way to lock the cells into a specific cell fate by modifying their epigenetics.
Sure. No, these are very interesting questions. And again, I try to explain it as in hopefully as simple terms as I can. So with regard to the single cell type, it always depends a bit like with what precision you look. We learn that more and more when we do single cell analysis. Now there might be always some variation, but in simple terms, we are very close to 100% of nerve cells, neurons. So that's pretty clear. And when the pathologists counted them, we got numbers of 98, 99% that we actually had. So there's a very small proportion of what are called glial cells, support cells, that are also there. And actually subset of those glial cells, they very, very slowly can divide, which is the normal function in the brain. And so, but nearly all of them are basically nerve cells. Now, not 100% of them are dopamine cells, but a significant proportion, obviously. So there are also subtypes of dopamine cells. And again, happy to go for the experts into those more details. That's again, some of the tweaks that in the future, we might think we can make them even more potent. But the first measures, they are all definitely nerve cells that basically hook up within the brain. The second part is, yes, you could probably do some of those steps. And again, in the lab, we are testing various ways to further improve that. Now we think from what we have seen, actually, once these cells are nerve cells, they are neurons, they don't divide anymore, they actually seem to be fairly locked into their fate. So we are not too worried about that they would suddenly switch into another cell type, but where we would like to use some of those tricks is maybe make the cells function even quicker. So if you looked carefully in some of our studies, it took up to 3 years for those, or we think it's going to take up to 3 years for the patient to get the full benefit. Why does it take so long? Can we speed it up? And so we have some ideas, again, from the lab side of tricks we maybe can bring to the clinic so the patient doesn't need to be that patient to actually get the benefit. And so that's something where we think some of those CRISPR techniques might be useful. And also maybe to even further protect or even treat the cells. So you can make basically super cells, now that maybe would actually, because when we put new cells in, seems to be great, we put a new set of cells that hopefully going to last for many years, but we don't really protect the other cells in the brain. The disease might still go on, and I alluded to that a few ways. Maybe what you can do, you can put in cells that are CRISPR engineered that also affect the disease progression. Again, not ready for prime time, but these are some of the ways you might want to use CRISPR technology in the future.
Again, I think a really comprehensive and fantastic answer. And that's the advance of these technologies, as we talk about in our community lecture series quite a lot. It's not always clear, right? When you develop PCR, when CRISPR comes online, what the long-term implications of that are going to be. So of course, these things get co-opted into basic science first, but you can see that the potential to be able to integrate into the development of clinical therapies is really tremendous. Pranav Sankar, who I am pretty sure is a high school student here in the Stem Cell Research Center, says that he remembers that you had talked about being able to activate or inactivate transplanted stem cells in the mouse using optogenetics. Could you elaborate how exactly that's achieved and what specific mechanisms allow that functionally?
Sure, that's a very interesting question again. And the way we really use this activation technique is more with the scientist hat, not so much we want to do that in the patient, at least not anytime soon, but more to try to figure out how do the cells really function. Because again, there was always a bit of question when you put stem cells in, do they just do something magic to the brain and then the brain is better? Or can we really see how they actually fire in the brain? And when we can do that, it's called an optogenetic switch. And again, it gets a bit into science slang, but it's basically you modify the cells such that they have now a way that they respond to light. So when you shine light of them, you can engineer it such that when they see light, you put actually fiber into the brain of the animal on the top, and the light goes through the brain to the cell. When they see the light, you can engineer it such that they just stop firing. They're completely silenced. But you can also do the opposite. You can engineer them when you put them in, they start firing like crazy. And so you can now figure out exactly how is the activity of the nerve cell correlated to the animal's behavior when it runs around, when it behaves, when it has no longer Parkinson's symptoms. So that's the area we think is very interesting to really basically test it. Now, theoretically, there were some discussions now, if you had these side effects, I mentioned at some point, some patients got uncontrolled movements with the fetal cells. Some people actually proposed maybe we could put in such a switch also in our cells. And then if suddenly the patients would have side effects, maybe not with light, but there's some other combinations of that where you can do it with a drug. You can actually, with a drug, stop them from firing. So it's, again, a very interesting question. And again, maybe in the future we come up with creative ideas. We need high school students to think about that, to maybe make even more powerful cells to modulate them with such techniques.
Yep. And again, that's, that's the next generation in this long process of developing, first understanding the system, and then being able to develop cells and cell therapies and regenerative medicine products with relevance. And it's one reason that the FDA, of course, is quite hesitant. And, this was all paving new ground in terms of moving forward with cell therapeutics. Early on, there was a lot of discussion about kill switches or ways to engineer in an off switch for any cell therapeutic, because we can't just stop it or take it back out again. These are really creative discussions about how one might take that to the next level. David, I apologize to this name, Kisalius asks, thanks for an engaging talk. What are the limitations to enabling diffusion, integration, and activation of the cells that you're transplanting into the brain? And so I think this is a question about cell delivery. Can you really get them all the places that they need to go in the numbers that you need from a therapeutic perspective?
Yeah, it's again a very, very important question. We thought quite a bit about that, because the cells need to basically reach a pretty large area in the human brain. And so that's why we need to make 3 needle tracks on each side, 3 deposits, because the cells actually don't move. So they stay exactly where we put them. They spread out a little bit like I made the point, but they stay there. And so, but actually what helps us is it's actually not so much where the cell body is, it's where they project the dopamine fibers. That's really the key. And they can do that over really long distances. And so we can actually calculate how far they can do it. And therefore, we can kind of mathematically calculate how far apart we need to put them to eventually reinnervate the whole region we want to reinnervate. So that's one area that we kind of thought about it quite a bit. And again, if you have time, there is even a more complicated argument. Right now, where we put the cells is the area that needs the dopamine, which is called the striatum. But actually, these cells, normally their cell body is not really sitting there. It sits in the substantia nigra that I mentioned. And from that, they have to project the fibers for about 2 inches in length. So a very long process. It's again something for the future that we are working on. We think maybe if we put the cells eventually into the midbrain, they might be even more beneficial. And there maybe again, this delivery of the cells and how they actually behave and integrate is yet another challenge. And maybe there again, the potency, the subtypes might be even more important. But yeah, it's a very, very important consideration to really make that work. Many therapies fail. They have maybe perfectly functioning approaches, but we don't know how to deliver the cells properly. So it's really a key component.
Yep, absolutely true. I think for many of us, certainly work in the CNS. A related question that's come into this is from Robert Clark, who asks kind of what could be considered a follow-on here. Can you discuss some of the volume issues that are related for injecting cells? And the specific issue that he's highlighting is, of course, the huge difference in size that you have between rodent models in terms of the preclinical data that you've developed and what's effectively going to happen in the clinical trial in the human brain.
Yeah, exactly. I mean, so that's in part why we did some modeling also in monkey studies. We did not use any monkeys for actual efficacy studies. And again, that's a more philosophical thing between different groups, whether that's really required or not. It's not really required by the regulatory agencies here. But so they can help us a little bit know with regard to the larger brain size, see how the cells behave in a larger brain and so forth. But what we found is that the cells actually, again, scale reasonably well, meaning that you can predict the length of projections in this context. The only difference is really that you need to make more sites. Nearly what I mentioned in the last question, you cannot just do it with one injection. So in the mouse or in the rat, a single injection can do it, can completely reinnervate the whole brain. If you remember, the rat that I showed you, one that was completely blank, the other one was brown. Below, they were both brown. So one injection site can make everything brown again. In humans, that wouldn't be enough. So on each side, we need 3 times 3, basically spots, so at least 9 spots to achieve that. But still, it's still a doable number, and we can calculate that to really reach the whole region that we need to reach.
Excellent. We now turn to a discussion of neuroimmune interactions a little bit. Tom Lane, who's one of our faculty here from UCI, says, wonderful talk. Thank you very much. And he's asking whether these cells, your cells, express MHC class 1 in response to stimulation with inflammatory cytokines. And for the audience, I'll just give a little perspective on that. This comes to an issue of how prone these cells might be to immunorejection, because MHC class 1 is something that's very important in terms of recognition of self versus not self, so foreign antigens. So Lorenz, please.
Yeah, so they definitely can express MHC class 1 proteins, and it's actually kind of in a much, you often do it in a maturation dependent way. We even studied that in collaboration, and for the scientists was guy Dave Seltzer at Columbia, who's one of the leaders in looking at inflammation immune response on Parkinson's disease. Even shows some of the dopamines that are particularly getting recognized by the immune system in this context might therefore be particularly vulnerable. And so there is, again, evidence that they can do that. And so there's, again, there was a lot of discussion with neuroimmunologists, what should we do about that? And in which context does it happen? And again, the argument that we probably should avoid triggering a big inflammatory response initially, because that releases all the cytokines and might trigger the response, make them more vulnerable. And again, the conclusion was that we want to do the surgery as careful as possible to not trigger inflammation, and do it in a way where we have immune suppression at least for the initial 12 months. And then again, the rest is really more empirical. We know that the cells can survive for up to 20 years. So we think it's definitely consideration. And we also know how we can suppress it. Now we have made a number of different strategies. And if I say by we, put on my Blue Rock hat. So there's a number of these universal technologies, bunch of different strategies, from don't eat me signal, they are called, to blocking MHC class 1 expression that are being tested, where again, you could avoid that completely. On the other hand, there is interesting studies from groups at Stanford saying some of those molecules might actually be important for some of synaptic connectivity in the brain. So you have to be quite careful fiddling around with some of those tools. But again, a very important question, and we don't really have all the answers yet. And a lot of it is kind of something we maybe need to have, basically, maybe the patients need to teach us ultimately, what works or doesn't work in this context in the long run. Yeah.
It's going to be an incremental process, that's for sure. Nicholas, if you're still out there, I apologize to you, but I think that Lorenz just captured part of your question right there, so we're going to pass by. Similarly, we have a question about injection site that I think you've largely covered. James Stewart asks, are dopamine neurons protected by myelin? And if so, how are they being myelinated? And are you going to be able to measure the success of that myelination if it's important?
So fortunately, that's not a big problem. So they're normally not myelinated neurons. And so in this case, that's not really a big issue. They're not very fast neurons. They're more neuromodulatory. And so again, they have this very unique morphology. I showed you one mouse neuron somewhere where they make this one process, and then they really innervate a large portion of the striatum. And I think all these features actually help us to make this as a particularly good target for cell therapy, because this is not a major concern as it might be for, let's say, cells you put in the cortex that need to go down to the spinal cord, where this might be much more of an issue.
Terrific. We have a question from Rick Robertson that, with apologies, I'm going to modify a little bit to broaden out. So he notes that this is really fascinating work with great potential. The embedded sense of his question is, as we have seen, these are my words now, for some regenerative medicine or really advanced therapeutics coming down the pipeline in recent years, these have ended up being out of reach, practically speaking, for a majority of patients, right? And so as promising as this looks, is there some concern, and how do we grapple with that? You as an investigator, we as a field in terms of stem cell biology, with to make these therapies broadly available to the people that need them most, as opposed to a select few.
I mean, I think that's, again, I said about a dozen times in my talk, the word off the shelf, maybe nearly the level to annoy you guys, but that was for me from the beginning part of the design. So it's always exciting to get a cell product to a phase 1 trial. Many scientists now dream to get finally there. But it's important that the phase 1 shouldn't be the end of it. So you need to design it such that it can be broadly applicable. And that was really the point, that we can make the cells at the scale where they could be broadly applicable to many patients. And at least from a pure manufacturing perspective, that could be done quite cheaply. So the cost of goods, like how much it costs to make one dose, is actually not very big. Now, what that means for the actual cost of the cell therapy might be a different question now, because we know that from gene therapy, making AAV vectors, the cost of goods is not very big, but the cost of the treatment is very, very different. So it depends how really that gets measured. And I think that should be a discussion in society, what is really a proper way to charge and make this therapy available to the broader community. What we can do from the science side is such that it's not already so expensive that's out of reach in the first place. And I have many colleagues at Sloan Kettering as a cancer center now, where they developed some of the CAR T cell therapies. And even if your best efforts, the cost of making the cells for each patient individually has been close to $500,000 in some cases. And so then obviously you can never make it affordable for the broader community. And so I think that's why in this case, that's not the limitation. I think the limitation is more going to be, how that's going to be negotiated and done, what's going to be the profit margin, the investment that the companies have to make up front. And again, I will definitely do my best to make it as broadly applicable and affordable to a broader community, because I think that's going to be essential. That also eventually can be done in other countries. Now that's, again, the frozen cells that can be distributed, very, very important. Actually, now we are working, right now it's still a little bit of a super expertise to get the cells properly needle. I'm very happy that my wife is a neurosurgeon who has done these cells for 20 years, knows exactly how to do it. It's not ready for any neurosurgeon to do it routinely, because again, it's a little bit cumbersome. But obviously that's what you're working on, that you can load the cells much more easily. You have a very simple device. And so once you have that, now you can ship the cells to a neurosurgeon, can do it in a frozen way. They can take the cells, now to inject, can be done in any country, hopefully at a reasonable cost. So that's the vision. Now, that the vision is going to go down that road, I hope not, but at least the vision is at least theoretically possible, because it was built into design from the beginning, I think.
Yeah, I think those are really just excellent observations, because it's the change in technology, right? I mean, whether it's a TV or anything else, it starts high and it comes down over time, and it comes down because of pressures from competing companies, from competing technologies, from the option to be able to do things elsewhere. Of course, designing in scalability from the beginning is really critically important. Again, just for our broader audience, because we do have a lot of community members here, this is something that California Institute for Regenerative Medicine, in addition to all of us as researchers and companies on their side, have really grappled with over the last 10 years or more, right? How are we going to address these challenges in terms of health disparities? It's interesting, some of the differences between NISTEM and CIRM. This is actually baked in part to the ballot initiative that refunded CIRM here. How that is going to play out, I think, is challenging, because there isn't anything that's prescribed. There isn't a pathway. But I think it's an important start that people are thinking about it from the ground up, as you've highlighted. So we have maybe two more questions here, if you'll bear with me, because we still have 97 plus participants online, and I can't actually see all of them. Some of them are on the YouTube and Facebook channels. So one is from Avi Tyagi. How is it that you can prevent graft-induced dyskinesias? And I think you had alluded to, I know you spoke about earlier today, this being an issue in terms of the fetal grafts. And so it may be purity of the population, but if you could just say a few words about that.
Yeah, so it's a very important question. Again, ultimately, it's again something we have to learn from the patient. Ultimately, we can do everything in our hands trying to avoid the causes we think were the cause in fetal grafts. And the common, most widely cited cause from studies in Sweden by Anders Björklund and others is the claim that in fetal cells, there were a lot of serotonin nerve cells, not only dopamine. And those cells can behave quite similar. They can take up this drug, L-DOPA, can convert it, but they don't have the right way to release it. So they release it in an unusual way. And it was shown with some drugs that affect serotonin, that in a patient that had the dyskinesia, they could be suppressed when they give the serotonergic drug. So that's some of the arguments saying that maybe that's the cause. And the good news is, no, we don't have those cells in our cells, unlike fetal grafts. we know that we don't get those cells. So that we can avoid completely. Now, is it really for 100% the only cause? We don't know. We think it could also be that the dopamine cells are not perfectly distributed within the brain. Actually, there are a few people that asked that question, know how they are spread out. And there was actually a case to be made in some of those fetal studies. They used a special technique. It's called like a noodle technique, where the cells were kind of like a string of cells, and they didn't really perfectly disperse. So they made this kind of what's called hot zones, where they had a lot of dopamine, but then other areas where there was nearly no dopamine. So the idea is if there's a lot of dopamine in one spot, that might trigger dyskinesia in this context. It might be abnormal movement. So that's again something we try to avoid by as well as possible spread out those nerve cells. And then finally, what we also learned from the fetal studies is that actually the patients that developed those side effects, if you go back, nearly all of them had already a little bit, or sometimes actually quite severe, versions of those side effects when they took the levodopa drug. So you can actually kind of predict by patient selection that they were already vulnerable for those side effects. Now it's no longer the drug, it's the cells that maybe does it. And so we want to avoid those patients that have major drug-induced dyskinesia in our cohort. And that should give us, again, additional safety margin to hopefully not see those side effects in our patients.
Yeah, that's terrific context. And it actually flows right into our last question for this evening, which kind of boiling down a little bit, the commentary here is whether these cells, the development of cells that you've so elegantly illustrated tonight, can those be used themselves in terms of investigating mechanisms of therapeutic resistance, in terms of investigations of the genomics or patient predispositions that could yield either preventative treatments or treatments that would impact, not just movement disorders in the future?
Sure. No, the answer is a big yes. That's something, again, we are very interested in our lab. We actually got a large award from an organization called ASAP, which is Aligning Science Across Parkinson's Disease, which is
Great acronym.
Exactly. They actually are ASAP. They move very fast. They invested nearly across like 20 groups or so in the initial effort, nearly, I think, $150 million, so a huge amount of money. And so specifically going again after, not all is for stem cell based understanding of Parkinson's disease, but it goes into the mechanism. And so that's something we think is absolutely crucial. And there's a lot of interesting ideas that we developed. For example, I'm leading a consortium exactly on this question, where we try to use stem cells in combination with machine learning to actually see whether we can use machine learning algorithms from patient data, genetic data, to stratify patients into different subgroups and basically recreate that in our stem cells and get additional information from them to see whether we can maybe find out treatments that would work in a certain subset of patients and match those treatments in such a way. But again, there's many different ways that is done by our group and many other groups. And just to give actually maybe a plug here at the very end for people who are really interested in that, there's a discussion not by NICE-STEM, but by NICE-CEF by the end of this week, where I'm actually also participating in a panel where this is going to be a major part of the discussion together with our collaborator on that big consortium project from Vic Corana from Harvard University and a bunch of other people. So people are interested, they definitely can look that up or contact me per an email. I'm happy to link them up to that NICE opportunity where this is further discussed as well.
Well, on that note, you actually, I can tell you, hit every button to get Pranav, our high school student, all excited again, because machine learning algorithms in medicine is his game plan. So on that note, I just want to thank you for really a wonderful lecture, for your patience in answering so many questions with such great detailed and easy to understand answers. Thanks to the administrative staff from the center, Judy Beck, Sam Johnson, and also to UCI Media, Will Alvarez. Thank you, Claire, for being here tonight. And thanks to all of our participants who really have engaged now for, I guess, about an hour and a half. And thanks for a great lecture, and have a good evening. Stay safe, everyone.